I'm José Antonio Carrillo de La Plata. I'm professor of the analysis of non-linear partial differential equations at the University of Oxford. And we are here at Queen's College, where I usually meet my students and I do research in this area. I'm going to explain you several examples of partial differential equations, and I will try to convey the use of these tools in the sciences, in particular in physics and engineering and in life sciences, where they find lots of applications. A differential equation is just the mathematical way in which we can write the relation between different magnitudes. The basic differential equation that we can say is position divided by time equals velocity. But normally we write it in mathematics in a continuous way. We assume that we have a path followed by a particle, which is a continuous movement, let's say, along this path in time, and we call that the position at each time of the particle. And then to write the continuous version of it, we just say that the rate of change in time of the position is equal to the velocity. In order to write that in a precise way, we say the derivative of x with respect to time equals to velocity. Here we are assuming that the velocity is given, but most likely is not known. For instance, the movement of each of the particles in the air filling this room is very complicated, and it can be very complicated even if, for instance, we are outside in the atmosphere. But anyhow, we can talk about that as a differential equation, and then the second law of Newton in the Principia Mathematica told us a long time ago that mass times acceleration is equal to the forces acting on the particle. And that we can write again as a mathematical law. We can write that the second derivative in time of the position is equal to the forces acting on the particle. Or, if you want, the derivative of the velocity in time is equal to the forces acting on the particle. That's the way in which we can write mathematically the second law of Newton in the Principia Mathematica. A partial differential equation is just an advanced tool related to a differential equation that allows me to write now an evolution for an ensemble of particles. So let's assume that we want to follow the movement of all the particles that are filling the air in this room. There are plenty of them. We know by the Avogadro number that there will be of order of 10 to the 23 molecules filling the air in this room. So we cannot talk about the movement of each single molecule that is filling this room because it will be hugely complicated. However, we can follow the density of particles in this room. Then we define a magnitude, which is the density at this position x in this room, and at each time t, that gives me the density of particles in the room, or if you want, the density of air in this room. Then we can talk about the evolution of that density. That is what a partial differential equation will give you. These kind of equations are typical in continuum mechanics, and we talk about continuous media because we follow continuously the density of that set of particles. And I will show you some examples of partial differential equations later on my computer. Some examples will be more historic because they have been important for the development of the theory of this field, and others because of the practical implications that they have. And they are the basic tools, the basic laws in which we can write all kind of mathematical laws used in physics, in engineering, in biology, in the life sciences, and lately they are encountering more and more applications in data science to develop methods to solve problems of optimization, machine learning, artificial intelligence, and many others. So, this is the Math Institute at the University of Oxford. Welcome. I'm going to show you my office. As you can see, it's a fantastic building, very beautiful. It looks very much like, almost like a Harry Potter movie now. The moving stairs. Okay, please come in. Let me then explain you what is a basic partial differential equation. Let me start with differential equations. Probably the most well-known example of a differential equation comes from Newton's second law, written in the Principia Mathematica centuries ago. So what it says, the Newton's law? So it says that if you know the forces acting on a particle, you can write the movement of that particle by writing the relation that m times the acceleration is equals to the forces acting on the particle. So this means that if I have a path which I start from some location x naught, and this particle suggests to those forces, I know the movement at each time of that particle that I denote by x of t, finding the solution of this differential equation. Why this is an equation? Because this x dot dot, what it means is the second derivative. This means the second derivative of x with respect to time. So the rate of change of the velocity in time, which is the acceleration, times the constant, which is given by the mass, is equal to the forces. By finding the location x of t, we are solving the differential equation and then finding the movement of the particles. Let me give you an example using numerical simulations of differential and partial differential equations. This is an example of a system of differential equations in which we are following the position and the velocity of a system of m particles, which are interacting through these different parameters and potentials. And they resemble Newton's like equation in the sense that they are based on giving you the acceleration of each of the particles. So this is a model that appears in collective behavior of individuals in which you want to obtain what is called the long time asymptotics of the system of particles, so meaning the behavior when time goes on of the system. And here I'm going to give you two examples of the behavior that you can expect. So here the arrows are the velocities of the particles, the little circles here are the positions of the particles, and you will see very quickly in this simulation here that the particles will arrive to a consensus in velocity, meaning that the velocity of all the particles, the arrows, becomes the same, and they are just then moving translationally in this direction. You don't see the movement of the particles because we center the simulation on the center of the particles, on the center of mass of the particles, so then you don't see really the real movement that is indicated by the arrows. Here in the simulation, it seems that all the time that I've been talking, there is no any kind of order of or some kind of typical behavior. It seems that they are just moving randomly around that location, like I don't know, like flies around certain location. But you will see that after a while, some behavior appears, and a kind of rotating behavior around certain center point becomes dominant, and you get this kind of behavior that we usually refer as the milling profile. So, in general, in differential equations, in system of differential equations, we are interested in explaining some of these behaviors and the generic behaviors that are stable, that appear naturally in the evolution of the system. Let me then show you another example of what is a partial differential equation. As I said before, I'm going to show you an example of what is a partial differential equation and what is the use of them by numerical simulations. Here I have a system of partial differential equations which comes from mathematical biology, in which U and V represents a density of cells. Let's say U is the one that here is pigmented in red, V is the one that is pigmented in green, and here I'm going to show you an experiment of what is called differential addition. You see the size of the arrows here, they represent how strong are the forces between the different type of cells. Here essentially the reds and the greens, they attract each other and between themselves with the same strength, while here the red and the green, they are attracting strongly than between themselves. So this leads to different behavior when the cells meet because of the different proteins that they produce and the different forces by addition that they exert onto each other. And here you have an experiment done in the lab with these different cells, that they have these different cell addition forces. And here you have our model based on those partial differential equations. And then you see the evolution, and when they meet, you see in this case that they produce a perfect kind of propagation front, or front in between the two species, while here they have an area of mixing that in which the reds and the green are somehow intermingling between each other, and the model somehow is recovering that size, the size of that intermingling zone. These differential addition forces can lead to different behavior when time goes on, as you can see in these simulations. in which you see that depending on the different addition forces between the two, then you recover either the mixing between the two species or somehow the one species surrounds the other at a certain extent, or even partially, like in this case, or they are totally segregated. So this is just an example that I hope you have enjoyed and you have learned now what is about the research in differential and partial differential equations. Thank you for listening.
First of all, I would like to thank the Spanish Embassy for giving me the opportunity to give you this talk. Today I'm gonna give you a brief overview about vaccine development against malaria, and also I will briefly talk about our latest results at the Jenner Institute at the University of Oxford. I am David Pulido Gómez. So let's start talking about malaria. So malaria is a human disease. This is transmitted by the bite of the female Anopheles mosquito. This disease is caused by the intracellular Plasmodium parasites. Of more than 120 Plasmodium species, only 5, those that show here in the table on the right, are known to infect humans. Of these different species, the deadliest of them all is Plasmodium falciparum. Plasmodium falciparum has a complex life cycle. It has two hosts, the mosquito and also the human. And in the human it has two different stages, the liver stage and the blood stage. So during the blood meal, the mosquito inoculates sporozoites that infect hepatocytes, liver cells. Then the parasites undergoes the periodicitic liver stage where it replicates, and this lasts between 1 to 2 weeks, and then releases merozoites into the blood stage. Then into the blood, So then you start the blood stage. During the blood stage, the merozoites infect the red blood cells, and then there is the asexual cycle where they replicate, and a subpopulation of those can transform or switch into the sexual development, forming or producing gametocytes. Then the mosquito, again during the blood meal, uptakes these gametocytes, and they can fuse, the female and the male, they can fuse, forming a zygote, and then this zygote transforms into an ookinete, abandoning the midgut of the mosquito, and there oocyst produces sporozoites that migrate to the salivary glands, and then the mosquito can infect again humans, releasing the sporozoites, and the cycle starts over again. Regarding the distribution or the global distribution of malaria, this disease is found in 91 countries worldwide. Here I show a map where you can see the annual global incidence. Darker color means a higher presence of the malaria disease, and lighter colors implies less of an incidence. As you can see, malaria is widespread in the equator, from America to Asia. In 2018, the World Health Organization reported an estimated of 228 million cases of malaria, just in that year, of which 213 and 93% occurred in Africa. Globally, every year is estimated to be more than 400,000 deaths. 85% of those deaths occurred in Africa and India. Sadly, 67% of those deaths are estimated to be in children under the age of 5. Malaria kills a child under the age of 5 every 2 minutes. Among the other people that are at risk of severe malaria are pregnant women, immunocompromised patients, and travelers. Despite all of this, international researchers, institutions, organizations, governments, and pharmaceutical companies are working hard trying to tackle this disease and eliminate and eradicate malaria. And if so, then the graphs I show you here from 2000 until 2015, both cases, as I show in the left, they're decreasing over time as well as deaths. However, if we want to eradicate, completely eliminate this disease within the 21st century, novel strategies will be required. And if so, the scientific community are developing novel technologies to tackle this disease. These promising novel technologies that are emerging include new diagnostics and medicines, especially those trying to tackle the resistance of the parasite, as well as vector control, new insecticides and chemicals for the nets or for spraying walls in houses, as well as passive immunization therapies such as monoclonal antibodies. Last but not least, there are vaccines, and the international community agrees that the development of a highly effective and durable vaccine would be key in the elimination and eradication of the disease. But what about vaccines on malaria? To date, only one malaria vaccine has been successfully developed. This one is RTSS. In 2015, RTSS with the adjuvant AS01 vaccine was approved by the European Medicine Agency for the prevention of Plasmodium falciparum in young children. This vaccine is a virus-like particle based vaccine where the circumsporozoid protein is fused to the hepatitis B surface antigen. You have the circumsporozoid protein and hepatitis B that forms a virus-like particle. Results from the phase 3 trials in Africa show that 3 doses given over 18 months provided a 46% protection from clinical malaria in children aged 5 to 17 months, with a 4th booster dose given at 20 months providing 36% protection over 4 years. This vaccine exhibits moderate to low efficacy, partly due to the high antigenic variation. So if we really want to eradicate malaria, we'll have to improve our current vaccines, and the Malaria Vaccine Technology Roadmap to 2030 calls to develop a next generation of vaccines to achieve 75% efficacy or more over 2 years against Plasmodium falciparum or Plasmodium vivax. So we need to develop novel vaccines, and the international community has not been idle about this subject. So lots of different research have been developed over the last 20 years, developing novel vaccines that target the different stages of the cycle of Plasmodium falciparum or Plasmodium vivax. I would like to give you an overview of the different vaccines that they are currently in clinical trials, have been tested in humans. So here I show a list of them that they tackle different stages, and I will explain them briefly in a second. So the first one of these strategies is similar to the vaccine RTSS that I just explained. They inhibit the sporozoids to infect the hepatocytes in the liver. Another strategy to develop vaccines mediate T cells in order to kill the infected hepatocytes. And you have these different vaccines that they aim to do that. Another strategy in the blood stage they elicit antibodies that they block the merozoite, the parasite, to infect or enter the red blood cells, preventing the malaria clinical symptoms. Other type of malarias, they're targeted to the parasite-infected red blood cells. And then, finally, we have malaria vaccines aimed to the mosquito stages, aiming to block the transmission of the parasites from the mosquito back into humans. I would like to briefly talk today about our efforts at the Jenner Institute in generating novel blood stage vaccines. These aim to reduce the sexual replication rate and hence protecting the vaccinee against a severe disease. For more than 20 years, it's been very difficult to find a vaccine in the blood stage to block the merozoite, here on the left, to infect the erythrocyte. And that has been proven a challenge because there are many different proteins at the surface of the merozoite that interact with receptors in the erythrocyte. On top of this complexity, another difficulty is that these different proteins are highly polymorphic and also they are redundant. If you tackle one, the parasite is able to use another one to infect the red blood cell. And it was until 2011 that the first highly conserved target was discovered. And this one was susceptible to vaccine-induced broadly neutralizing polyclonal antibodies. And this antigen is Rh5. Rh5 molecule on the parasite interact with the pathogen receptor, also known as CD147, at the surface of the erythrocyte. Rh5 protection based vaccine has been shown in animal models. Rh5 vaccines are able to protect mouse, rats, rabbits, and non-human primates. For the first time, a blood stage vaccine has shown in the first in-human Rh5 clinical trial a significant delay of the disease. This is great news, but however, we need to continue improving the current vaccine. In 2014, it was reported the Rh5 bound to pathogen structure and also neutralizing maps that reveal where the protective epitopes were, and by precision immunology we could modify the antigen, making it focusing towards these protective epitopes. Also, thermostabilization, introducing point mutations, performed a thermostable molecule, making it a better vaccine. Also, we have engineered this thermostable Rh5 molecule to conjugate it onto a hepatitis B surface antigen, forming a virus-like particle similar to the RTSS vaccine. Our Rh5 virus-like particle-based vaccine has shown an improve of 5 to 10-fold in animal models. And we're producing this vaccine to GMP standards and planning to evaluate the Rh5 VLP-based vaccine in the clinic next year. I would like to thank all the members of the group, past and present, as well as our collaborators, our funders, and thank you for listening.
Hello, everybody. I'm Andrés Jaramillo. I'm a professor at the University of Warwick, and I work on synthetic biology. The University of Warwick is located in the center of the UK, very close to the birthplace of Shakespeare, and it's ranked among the top UK universities. So the goal of our lab is to design some smart biological molecules to be used in therapy to cure diseases. Okay, so we want to create a new generation of these therapeutics that are able to differentiate between one target and the other, and very specifically. Okay, so if we can have this single cell specificity, then we'll be able to not only kill antibiotic-resistant bacteria, but also parasite that, like the plasmodium of producing the malaria disease, which is killing most people in the world. And we could also use it even to produce an antimicrobials that can just target cancer cells and not normal cells. Okay, so what you will avoid any secondary effects. Or even you could just target senescent cells. These are cells that are aging and older than other cells. And then that makes a whole tissue older. If you kill those, then the newborns occupy their place, and then the average age decreases. Okay, so then there is a lot of opportunities and a lot of applications if we are able to design an antimicrobial that is able to kill very specific cells, organism specific and antimicrobial. So, but then how are we doing that? We are doing that by using an automation of this design. So normally this by the name automation is used with computers and also with robots, and then people may get the misled that our automation is just using the computer to help us to design. Like you can use computers to help to design a car or to design some other things. Or you use a word processor to help you to write a manuscript. So then all this formatting is done automatically by the word processor. It's helping you. Yes, this is not with what we do. The equivalent would be that you tell the computer and to your word processor, please write me a book or manuscript about some subject, and I'll go to the beach, and then you do it for me. That automation of the creativity is what we are aiming for. Okay, so for that we have to simulate on the computer all the laws of physics for the molecules, and also how evolution works, because we use the same type of methodology that nature uses to produce complex matter like us, living organisms, and to use it to produce our multifunctional smart molecules. Therefore, this is the key thing, this automation of the design. But this is done only when we have small molecules like proteins and or single nucleic acids or mixtures of them, and we have published that throughout the years. But now we are aiming even bigger. We want to have these organism-specific and antimicrobials. We want to specifically kill cells. So for that, we need complexes, assemblies of proteins that makes a very complicated machinery. Okay, this is far beyond our knowledge on the physics, how to model the physics of that. Therefore, what we do is we use bioreactors with living cells, and we express those in molecules inside the cell, and then we use the same type of algorithms to accelerate the evolution we use in the computer, but experimentally. Therefore, instead of using some random number generator, we increase the mutation rate and with some chemical, etc. Okay, so that's how we do in the lab. A mimic of what the software does with this optimization of the design. And we test, of course, all our systems, either we do by directed evolution or in experimental evolution, or we do it in the computer. And we test it with in bioreactors, microbioreactors. Why micro? Because we want to see single cells. As you can see here, this is our bacteria. You could see at the beginning how they were just in replicating. And this is of the size of 1 square meter. No, sorry, one in a square centimeter. Okay, so this is the size of a coin. And you have more than 200 in chambers like this one, where you can have a monolayer of bacteria just growing, and you can all of them focus and maintain the focus because it's an automated microscope, and then you can just monitor the gene expression by using fluorescent reporters. Okay, so that's very important in order to know carefully how our systems behave, our multifunctional molecules. And then we also, as I said, we also aim to make these antimicrobials as assembly of proteins, and this is very useful to kill specific cells. So what we have done is to create a strategy where from the genome sequence of the cell we want to kill, we can know all the molecules that are around this pathogen, and then we don't need to culture the pathogen. We can use our methodology in order to create the antimicrobial that is going to kill the pathogen. Okay. And we create a cocktail the same way that the cocktails of antimicrobials are used against viruses like HIV, but in our case, it's against this pathogen to avoid the emergence of resistance due to mutations, because nature finds always their way, so we need to counteract that and minimize that. It doesn't mean that we are going to kill forever the resistance. We are going to minimize it quite a lot. So, so this complex of macromolecules, what we do is we take advantage of some complexes of macromolecules that are already available in nature that are the viruses. But these are viruses that only attack harmless bacteria. So we use that to evolve it in our own bioreactor. So you can see this, the pumps we have 3D printed and designed ourselves, and also the valves. These are all we use in all these CAD tools for 3D printing. And also we have we made a farm of bioreactors so we can evolve in parallel all the bacteria. Okay? But very importantly, once we have finished with this evolution, then we remove all the things from this virus, and we only get the proteins, and then are those that we use to kill. So this is just a vehicle in order to evolve faster, because viruses are the faster evolving entities on Earth. Okay, so then finally, we are also aiming to create even more complex systems, more smarter molecules, but these are smarter cells. Can we make bacteria to be a little bit more intelligent? How much intelligent? Imagine that we can make bacteria able to play board games. So that's exactly what we have done. We are engineering Escherichia coli, this is a commensal bacteria we have in our body, to be able to learn by themselves to play tricks. They can learn many things, even the alphabet, or they can learn to play tic-tac-toe. Okay? And importantly, they use the same mechanism than our brain to learn, reinforcement learning. So this is done by you. Once you can penalize and punish the cells with antibiotic each time they have lost the game. But you don't tell what was the wrong thing. It can be any of the moves that led to lose the game. Okay, and that is the key of reinforcement learning. Okay, so then they figure out, okay, and how we do that. We do that by mimicking neural networks, but with genetics and in bacteria. So let me just thank all my group you see here, and especially all of you for your attention. Thank you.
Hello, my name is Rocío Martínez Núñez, and I'm a principal investigator in the Department of Infectious Diseases at King's College London. And what I'm going to present to you is part of the work that I've been doing to increase diagnostics capacity in the UK and Spain. So our objective was to increase diagnostics. And our labs, before the pandemic hit, looked a little bit like this picture. We have plenty of reagents and plenty of equipment. However, what we found ourselves during the pandemic was a little bit like what happened in supermarkets. We did not have enough reagents, we did not have enough equipment, and therefore it was impossible to cope with the demand for tests. Therefore, we needed a solution that was fast and something that allowed us to increase diagnostics capacity. We needed something that was available immediately, that was cost effective, that was universal, so that many labs could use it. So how do we increase diagnostics capacity? Using automatization and also creating new protocols for detection of SARS-CoV-2. With regards to robotics, we needed something that was available then, that we could afford, and also that was flexible and usable by different users. And this is why we opted to go for a company called Opentrons, because these robots are open software and hardware, and therefore are customizable to the needs of every single lab. And also because they do not require a set protocol, the user programs them to do the protocols that are ongoing in the lab, and therefore they would be used by many different labs. The timeline, the calendar that I've been showing, is actually the timeline that it took us to bring the first 4 systems in Spain. So we started a group to basically gather our expertise and time on the 17th of March. And in April the 3rd is when the first 4 installations landed in Madrid, to go 2 to Barcelona and 2 to stay in Madrid. And the keys to this was coordination and compartmentalization of the task. We had an incredible team. We had really good communication, and each one of us took a task. We had to coordinate between different countries. The company is based in New York, I'm based in the UK, the rest of the team is based in Spain, and the robots are made in China. We had a lot of communication between us, and every time there was a problem, we simply saw it as an issue as to how to solve it, rather than a wall that we couldn't climb. So this led to the first 4 installations to be set in on the 3rd of April. And given the repercussions of these robots, we ended up bringing another 14 installations that are now in many places all around Spain. And this results in around 1 million tests per month extra. With regards to the new pipelines, we have been developing this in KillSet Partners, which is an academic health sciences center, and encompasses St. Thomas's Hospital, King's College Hospital, as well as Guy's Hospital and infectious diseases. And what we did was essentially create a new lab called the June Almeida Lab in honor of the first virology that discovered coronavirus. And she was actually employed in St. Thomas's Hospital. So here you see a very simplified flowchart of how CoV-2 is diagnosed. We first always need to inactivate the virus because the samples are potentially infectious. We then need to extract the genetic material of the virus, which we then need to detect. So what we have been developing in the June Almeida Lab is protocols of heat inactivation, we have been using a mix and match approach, which allows us to mix different reagents that we usually use in molecular biology experiments rather than in clinical diagnostics, validate them through NHS England, and also we have been incorporating a tracing app for our samples thanks to a collaboration with Professor Giralt in the University of Malaga. And what this allows us is to provide multiple solutions precisely to avoid running out of reagents. If we are able to use multiple reagents and multiple companies with the same efficiency, this allows us to have multiple pipelines and build resilience. So in here, I wanted to show you some of our results. And in the first top two, what you see is the detection of a virus using two different primer probes. And what you see is that different methods of extraction do not change and do not modify the result. The CT values, which is what we use to detect the viral material, are the same. And this is independent of the RT-PCR reagent. In the upper case, you see fast virus, and in the lower case, we use Luna. And the detection was similar amongst the samples. We also use different one-step qPCR master mixes from the three different companies. And what you see here is a graph where we assessed multiple donors. And as you see, the values of the PCRs did not change when using one master mix or another. The other thing that we wanted to investigate was if the primer probes combinations, the reagents we use to actually detect the virus, are equally efficient. And our first experiments told us that if we could use either the probes for the N gene or RDRP, were equally efficient. However, when we started using samples that had much lower viral loads, which means higher CTs, because you need to amplify more to be able to see it, what we saw is that the red dots, the RDRP gene, disappeared. So actually, it was the detection of N that allowed us to see samples that had very low viral loads, and we also assessed how heat could interfere with this process, and we saw that there was no variation in our qPCR results. We also investigated if other kits could observe the same, and in here you have some results where you see in blue the detection of N, and in green the detection of ORF1b, and actually for previously classified negative samples, the detection of N allowed us to be able to see virus where other platforms had deemed those samples negative. And in here, I just wanted to show you this graph because it really shows that detecting N using different reagents is very, very comparable. So it's not a question of the N reagents that we were using. Using different reagents renders the same result. And also we use different plate formats. And this is really important because usually in clinical diagnostics, we use 96 well formats, but in the lab, what we are using is 384 well plates, which allows us to assess 384 samples at the same time, saving a lot of time and reagents per extraction. The next thing that we need to understand is if these samples that have very low viral loads that we are able to detect with N are still infectious. And the last thing that I wanted to show you was that we have incorporated in our protocols heat inactivation, which we have seen, as you see on the left graph, that warming up samples at 70 degrees for half an hour, that does not modify the results of the PCRs. And on the right graph, you see very similar data using both positive and negative samples. So this is really important because this allows essentially to diagnose SARS-CoV-2 in labs that do not have Cat3 facilities, and essentially allows us to expand diagnostics in many, many different settings. So at the moment in the June Almeida lab, what we are doing is develop new protocols using our homebrew and homemade reagents. We are also evaluating saliva for testing because it's a much easier material to use than and avoids the nasopharyngeal swabs. And we are also working on new novel methodologies, including LAMP, including LAMPOR, We are also developing new inactivation protocols, always having in mind cost effectiveness and universality of use. And we are also investigating multiplexing because we are now getting into the autumn, and this will require us being able to distinguish different respiratory infections, which we think is now essential to be able to control the pandemic. And at last, from King's Health Partners, what we want to do is contribute to mass testing in the UK. At last but not least, I wanted to leave my slide of acknowledgements. I have a lot of people to thank. Every person here is a volunteer in these projects, and I would like to thank everyone in King's College London and King's Health Partners for making such an amazing team. And in Spain, everyone that has put their heart and soul on this project to install robots. Thank you very much.